So I'd now like to introduce you to our third speaker for the morning. It's Susan Galbraith. We're delighted to have her here. She's Vice President and Head of Oncology at iMed AstraZeneca. I'm sure none of you can have missed the news about AstraZeneca moving its global headquarters to Cambridge, staged over the next several years. And so without further ado, I'd like to introduce Susan to you. Thank you.
Thank you. Look at the slides up. Thank you very much. Okay, so you've already heard that in the UK we've got a great track record of medical innovation we can be proud of. We've got highly regarded centres for research, both basic and translational. And we've got a great track record of discovering and delivering medicines. But the truth is that the medical needs that exist globally are huge. They're considerable. And because of the demographic changes that we've got, overall, these are growing. So if we're going to be even more successful, make a difference to all of the patients that are bearing this burden of disease today, we're going to need to do even better in terms of our translational science. So we've had a new CEO at AstraZeneca who joined us at the end of last year, and his strategic imperatives that he's talked about are based very simply on following the science and putting the needs of patients first. And as a clinician scientist myself, those words resonate very strongly with me. So I'm delighted to have Pascal Soriot on board as our new CEO and to help deliver that strategic intent. So we're focusing our resources on a smaller number of core therapy areas in cardiovascular and metabolic disease, in respiratory and inflammation, and in oncology. And oncology is the area that I'm trained in, and I find it a very exciting point to be at in terms of cancer research because of the information that we're getting from the genomics revolution. I really think is starting to make a difference, and you are seeing drugs coming through where we've really put that information to task and understand which patients we might need to treat with our new medicines. So if we're going to be successful and more successful than we've been as a pharmaceutical industry in recent years, our strategy is then to focus on the quality of what it is that we're doing. And at AstraZeneca, we have a framework we call the 5 Rs to ensure that quality focus that we measure all of our programs up against as they're coming through development. From my perspective, it starts right from the beginning by understanding which are the right patients to treat. So if we understand that link between target and disease and get that right and choose the right target and the right patients to treat, I think the rest of it follows more reasonably. But we haven't done that well enough. And I think one of the core limitations to doing that well enough is not just understanding that a particular pathway might be important in a particular cell, but understanding in the prevalence of the disease, how frequently is that a core driver of the disease, and how important is the inhibition of that target and that pathway going to impact on that disease process. And the key limitation for me in my area to do that is getting access to human tissue human cancer tissue. So the limitation of access to tumour tissue, understanding that, profiling it, being able to use it to create the right kind of preclinical models has been a limiting factor in the understanding of the relevance of what we do in the lab and translating it through into the clinic. So I also think within the early clinical trials, we've got to continue to test that human target validation and make sure that we're actually inhibiting the target that we think we want to address adequately, that we understand the tissue exposure relationship to do that properly within the tumour tissue. And again, the limitations of technology and the limitations of access to tumour tissue have stopped us doing that as well as I think we need to if we're going to really be successful. If we understand that as well, we can better understand the impact of that target inhibition on the safety profile of the medicine and make sure that what we're actually delivering is something that is going to have an acceptable safety profile for the patient population we're aiming to treat. And finally, you'll see that right commercial is in there as well, but my view is that that comes after you've done the other pieces right. Good medicine is good business. But if you get it the other way around and you focus on the right commercial before you've worked out the other pieces, that is not a recipe for success. So I think in terms of how you take that approach to what we're trying to do and then put it together with how we work together with the other aspects of the knowledge-based cluster that you've talked about, it requires partnership between the industry, between academia, between industry and the clinicians that are treating patients every day as part of the National Health Service. We believe at AstraZeneca that the UK is well positioned to be a leader in translational research, and it's a major reason why we've announced that our new global headquarters will be here in the UK and right here in Cambridge. We are well positioned as a country, as you've heard. We do have great scientists. We understand about linking those scientists to the needs of patients, and we've got a really unique opportunity to leverage the infrastructure of the National Health Service, which is already well linked to our universities. We have an opportunity to connect our countrywide National Health Service patient data to the genetic information that we can generate now to help ensure we are putting the right patients on the right drugs and putting the right patients on the right clinical trials as we're trying to develop the next generation of drugs. In my area of oncology, that opportunity is particularly clear. The underlying science in oncology is showing that diseases that we used to think of as one disease, like lung cancer, are increasingly segmented, and each different segment has different genetic aberrations to which the cancer is addicted. So there's a great opportunity now to provide different experimental drugs to those different patients in those different segments. But to do that effectively, we need to have that linked up to that countrywide information. And so that we can link the patient that has the right genetic abnormality with the opportunity to go on a clinical trial for a drug where they're likely to be sensitive. So I just want to illustrate that just a little bit with the development of a drug called Iressa. This is an epidermal growth factor receptor inhibitor. And it turns out that in around 10% of lung cancer in the West and about 30% of lung cancer in the East, there's a mutation in the epidermal growth factor receptor which drives particular sensitivity to this. Now, this fact was discovered after the drug was already in late-phase clinical trials across non-small cell lung cancer, but the data that was published in this trial, IPAS, which was run predominantly in Asia, showed that if you look at the progression-free survival on the y-axis, if you look in the overall patient population, there's actually a crossover of the curves in green for gefitinib or Iressa and the standard doublet chemotherapy that's used in yellow. If you now separate those data out by those patients that have a mutation, so the solid lines in green for gefitinib and in yellow for chemotherapy, those are the patients that have a mutation that drives sensitivity to the epidermal growth factor receptor, and the dotted lines are the patients that don't have the mutation that are wild type. You can see that for patients treated with gefitinib, the difference between the solid line and the dotted line is large, whereas the difference for patients treated with chemotherapy is virtually the same in both with mutation or without a mutation. And these data help to lead to the approval of Iressa for the treatment of EGFR mutant non-small cell lung cancer, aligned with a personalised healthcare companion diagnostic for that EGFR mutation. And it's now approved in over 70 countries and prescribed alongside that diagnostic test. So, however, I think the story of Iressa is a story of how things could be better from my perspective, how we could have done the clinical trial development better, and how we could now be delivering care better, having got a drug and now understand which are the right patients to treat. In addition, when you're thinking about the next generation of drugs for lung cancer for the next segment, we can do more to improve the efficiency of those clinical trials and the probability of success. Right now, AstraZeneca is running a program with the potential to address less than 10% of lung cancer patients, and we're having to run a phase 3-sized screening effort to answer what is a phase 1 or 2 question about early clinical activity. If you just to think about this as a system, it doesn't serve any part of the people that are participating in that system well. From the patient's perspective, they're in the strange position of almost hoping that they've got a particular mutation, one that gives you actually a poorer prognostic outcome, so they can get access to the trial. That's an odd thing to be wishing for. From the investigator's perspective, they get screening fatigue. They have to screen so many patients to get one patient on trial. And from the perspective of the sponsor, we have to get a huge number of sites up and going so we can get access to the right number of patients. And the system is expensive, it takes too long, and it doesn't serve the patients well. So my proposal is, when you've got a system that's not working for anybody, perhaps we should think about changing it. A better system would be to have a genetic screening as routine at diagnosis, a genetic screening capability and informatics support to a common high standard countrywide, and the logistics links to enable access for multiple investigative centres to get access to multiple different experimental drugs, so most patients can access them, not just a few. The UK is in the rare position of having the National Health Service infrastructure, which, if we linked it to our genomic analysis capability and the right trial logistics and access to a range of experimental drugs, not just from a single pharmaceutical company, but across the industry, we could deliver this in a way that few other countries could. I'd certainly be happy to put the portfolio of cancer therapies for AstraZeneca as part of that, and not just in an exclusive way. So because we think this is important to change the paradigm, AstraZeneca is giving financial and scientific support to Cancer Research UK's Stratified Medicine Initiative, together with Pfizer and the government's Technology Strategy Board, to help create this kind of high-quality genetic screening infrastructure. Now, my family tell me that I'm impatient by nature, and it's a true observation. But I would say that what we're doing is still not enough, and we're taking too long. If we got this right, the potential difference it could make to patients is also illustrated by the fact that even after we've made the investment to get a drug developed in the right patient population and get it approved, the uptake of diagnostic testing in this country and across the EU is behind where it could be in the world. Right now, only half the patients that could get access to a drug like Iressa actually do get access to it because of the diagnostic testing is less. And we do know that it's possible to get that testing rate much higher than it is here, because it is being done in other countries. Creating a system for linking the genetic screening information to patient outcome has another benefit. It would also drive greater innovation. If we go back to those curves, even though you see a much improved progression-free survival in the patients that have got the EGFR mutation, one of the depressing things about this curve is it ends close to zero. Patients progress. And if you understand the mechanisms by which you get resistance to the current therapy, we can design better treatments. So we're already doing this. By having access to tissue at the time of progression on epidermal growth factor receptor inhibitors, we've learned as a community that about 50% of that acquired resistance is due to a secondary mutation, T790M and exon 20 of the epidermal growth factor receptor. And what that does is create a block to the entry of the drug into the pocket that you can see here. This is Iressa actually bound in wild-type epidermal growth factor receptor. So once we know the structural conformity of that newly mutated pocket, and we understand the difference between that and the wild-type receptor, we've got great chemists at AstraZeneca, and those chemists have designed a new medicine which binds selectively to the double mutant and binds selectively over the sensitivity of it to the wild-type receptor. So what that means is we hope we can get a better medicine that has greater activity both against the single mutated and the double mutated drug, and also a reduction in the side effects of skin rash and diarrhea that you get from inhibition of the wild-type receptor in the rest of your epidermis in your skin and your gut. Early clinical trials are ongoing with this new drug and are looking very encouraging. So I just want to move on. I think we could do better, more rapidly, if we understood that kind of resistance mechanism more broadly across different classes of drugs for cancer. And by that kind of collaboration, getting access to tissue and understanding the science, we'd also, through collaboration, enable us to develop the next generation of great scientists, because through the collaborations and discussions we have, we'll get better connected, more entrepreneurial scientists who thrive on seeking out those opportunities to solve the next problem. So what kind of collaborations have we done already at AstraZeneca? One example is collaboration we established in 2011 with the UK Medical Research Council. We provided access to 22 compounds to MRC researchers, and they've got their own list of potential hypotheses that they want to test. We have a similar deal agreed with the National Institutes of Health in the United States, and an innovative approach with the Karolinska Institute in the cardiovascular metabolic area. In the UK, we're also working with the NIHR's Cancer Research Network. The formation of experimental cancer medicine centres, which are 50-50 funded by the Department of Health and Cancer Research UK, has made a huge difference in how cancer research is organised and coordinated. And this has made a real difference to us at AstraZeneca. Reduces the time they get for contract negotiations and site set-up time, and it's restored the strength really of the UK as a phase 1-2 trial centre of excellence in oncology. From our perspective, what that means is we get more ideas tested, so more clinical trials with our drugs, with ideas that we hadn't thought of internally, and as one of my colleagues says, at least half the brilliant people in the world don't work at AstraZeneca. From the investigators network and from the patient's perspective, they're getting access to more novel drugs. So I think this is a win-win scenario, and we're very happy with the progress that's been made on this since it was set up in 2009. We also have a track record of working well with individual academic centres, and one example is the relationship we've built with the Manchester Cancer Research Centre. It works well because it's got mutually overlapping science, we've got good relationships, and we've got some staff interchange, and it helps that we're just up the road. So in 2006, we recognised some areas of mutual research interest, phase 1 clinical trials, blood-borne biomarkers, and imaging. And then we've added radiation oncology, tissue-based human target validation, and lung cancer more recently. We have a joint steering committee, quarterly reviews, so we make sure that work's completed according to metrics that were mutually agreed. And if we've got problems, and problems do come up, we work through them and we've managed to remove the blockers. So this week we've also announced 3 new collaborations with groups here in Cambridge, and I very much hope that we can build on the model and be just down the road in Cambridge, and that these collaborations are just the start of the kind of exchange and partnership that has been talked about by Sibylle and Spike, so that we can actually improve how things go. So we're very excited to be right at the heart of the Cambridge network. So this is the Cambridge Biomedical Campus, and this is the site where we're going to be located right next to the MRC, Laboratory of Molecular Biology, just across from Adam Brooks Hospital, and right next to the CRUK's Cambridge Institute. So our choice of site is grounded in the belief that collaboration's key, and that by being able to walk across and have a cup of coffee and lunch with guys working in these other institutions, that will make a difference to what we do. So what I'd like to say is I think there's a great opportunity. I think there's more that we can do. Very happy to be part of that dialogue about how we can work together and what we can contribute from the pharmaceutical industry to make sure that makes a difference. And I always remember that when I was treating patients with cancer, that I didn't have enough tools in the toolbox of what I wanted to do. And the reason why I joined the pharmaceutical industry is I wanted to be able to make more tools available so that physicians treating patients with cancer have more opportunity to treat them with something that's going to make a difference. So with that, I'd like to stop and answer any questions that you might have. Thank you.
I'll ask a very quick one, Susan. We've heard from Pascal that the chances are that there'll be up to 2,000 jobs created in Cambridge. Do you have any idea of the timescale for that?
So the intent is, it's interesting, I was hearing about the concept, I think you said for the Francis Crick was 2007, and you're going to be up and going in 2015. The concept that we've got is 2013, and we're going to be up and going by 2016. So timelines are tight. And in fact, what I also want to be able to do is get a forward party, perhaps located here in Cambridge, earlier than that, perhaps even as early as next year, if we can find the appropriate space. Obviously, our colleagues at MedImmune are already located at Granta Park, so having at least some additional office space down there for some of the AstraZeneca people is one option. We're discussing other options. Around, because I think that will be very helpful to help drive the collaborations in the meantime. It's about 1,100 people to move from Alderley Park down to Cambridge. That's not a small logistical exercise. But yeah, we're looking forward to getting started, and the building should be completed by 2016. That's the goal.
Okay, well, thank you very much indeed, Susan.
Thank you.